This conference will now be recorded.
Hello students. Now we are preparing for NEET 2024 and now we are moving to a new botany chapter, chapter number 10, cell cycle and cell division. So in the 10th chapter, actually this is a very important chapter, means you should understand this very clearly. Otherwise you will feel like questions are a bit confusing. So please try to learn this chapter thoroughly so that each and every question, read the question very clearly, because there will be some kind of confusing questions from this chapter, in the sense like 2 3 concepts they will mix, then they will ask the question. So such kind of questions will be creating some kind of pressure. Like, don't no need to get tensed. So you watch this clearly, and I'll try to give maximum number of questions from the previous question papers so that it will be clear for you. So let's start now. So in chapter 10, cell cycle and cell division, we are learning about different stages of cell division, or a cycle, the whole cycle of that cell, and different stages of that cell division also we are learning. So here in the NEET syllabus, in the 10th chapter, phases of cell cycle you need to learn. In the whole cell cycle, in which in all phases it is going through, that cycle you need to learn. Then about different cell division, like mitosis and the significance of mitosis, meiosis, stages of meiosis. In that meiosis itself, you have to learn meiosis 1 and meiosis 2, stage 1 and stage 2. Then the difference between mitosis and meiosis. Actually, this much only you need to learn in this chapter. But don't think it is a very simple chapter. You will get good questions, very good questions from this chapter. Okay, so let's move to the chapter now. Cell cycle, that's the first topic which we are going to discuss in this chapter. Cell cycle means, here the activities, all the activities of a cell, from one cell division to the next. Okay, all the activities, all the activities of a cell. Okay, that's what you're learning in a cell cycle. The whole activities of a cell from one cell division to the next. Okay, here you can see one cell, how it is getting prepared for a cell division and how it is undergoing cell division. And after that, it's forming a new cell, and that new cell is also getting prepared and moving to the cell division. So that cyclic process, that's what you are going to learn in this cell cycle. So there the cell duplicates, what duplicates? Genome duplication, then synthesis of cell constituents, then it is going for division. Right, when a cell is forming a daughter cell, it's dividing, means what and all that should be there in that newly formed cell. The genome should be there, then all other cell organelles. So that cell need to synthesize everything first, right? First the genome should get duplicated, then synthesis, all other cell components should be there, another copy. Then it move to, after that, after all these processes only, it will move to the divisional stage, right? So it's like a story, slowly it will grow, it will move to the other stage, right? So activities, all the activities of a cell from one cell division to the next. So through this, that cell will duplicate the genome, Then other cell constituents are getting synthesized in the cell, and slowly, eventually, that cell is moving into a divisional stage, or it is dividing into two daughter cells. Clear? And in a eukaryotic cell, you can see that cell cycle. In eukaryotic cell cycle, you can see mainly two stages. Okay, two main stages, which and all you can see. First one is the interphase. Okay, this is the first phase. Then mitotic phase. Second phase is the mitotic phase. So here in this picture, you can see that here one stage is there. You can see this mark, right? This forms the interphase, and this is the divisional phase. Here the division is taking place, right? So in the this interphase and mitotic phase, interphase is called, we can mark it as I phase, interphase, and mitotic phase or the divisional phase, M phase. Okay, so here the eukaryotic cell cycle that is having two main stages, that is interphase, or that is the preparation phase, and mitotic phase, that is the division phase. So a cell's progress through the cell cycle, that is regulated by some proteins. Okay, the cell's progress, that means progress in the sense the cell cycle process. See, there is a cyclic process. So from a daughter cell, that daughter cell is maturing, or it is going through, the cell growth is taking place, genome replication is taking place, then that's somehow it is getting prepared for the mitosis. So each stage, that progressing of each stage, that is controlled by some proteins, or that is regulated by, we can say, cyclins. Cyclins and cyclin dependent protein kinase. Cyclin dependent protein kinase. Kinases. So we can write in short form CDKs. Okay, cyclin dependent protein kinases. Okay, so the cells progress through different stages, that is regulated by cyclins and cyclin dependent protein kinases. So cyclins and cyclins dependent protein kinases. Okay, so let's see the interphase. Here two stages we need to discuss. First is the interphase or the preparatory phase, and next one is the divisional phase. So let's see the interphase first. Here interphase in this diagram, see this diagram you need to learn very clearly. Don't forget, you have to keep this in your mind so you can answer many questions. See, in this we have marked G1, S, G2 and all, and here we have marked the division, right? Up to that there is no division, and here you can see inside we have given a mark. This is the interphase or I phase, right? Interphase, and here only this little portion we have given or marked as M phase. Clear? So interphase, we can easily say that this is the longest phase. Okay, this is the longest phase. Which one? Interphase. Interphase is the longest phase, and around 96% of the cell cycle it covers. Here from this diagram it's very clear, right? from that figure. So 96% of cell cycle is covered by the interphase. And we said this is the longest phase of cell cycle, and during this phase you can see here what and all happening here. Cell growth is marked, DNA replication is marked, preparation for mitosis, that is also marked. So this, during this time, this uh, cell is getting prepared itself for both cell growth and DNA replication. Okay, this is the preparation stage. So during this preparation stage, here cell growth is taking place, right? Cell growth is happening. Then DNA replication. DNA replication. And finally, that is getting prepared for mitosis. Preparation for mitosis, or it is getting prepared for the cell division. So this is the longest phase and it covers around 96% of cell cycle, and during this state the cell is getting prepared itself for both cell growth, then DNA replication, and it is finally getting ready for the mitosis. Okay, so this phase is called as the preparation stage or preparation phase. Okay, and this consists of the interphase consists of what and all? Interphase consists of G1 or gap phase 1. G1, or we can call as gap phase, or we can say post mitotic gap. This is post mitotic gap. Post mitotic means here mitotic division over. So post mitotic, after mitotic division, a gap is there. Gap for what? Gap to the next mitosis. So that gap, post mitotic phase or post mitotic gap. Then here comes the S phase or synthesis. Synthesis, or this is the synthesis phase or S phase. And here G2, G2 or gap 2. This is gap phase 2. Okay, or this is pre-mitotic, pre-mitotic gap phase. Okay, here you should understand post-mitotic gap phase, which is G1. Post-mitotic, here one division, one mitotic division got over, and this is a gap after that. Okay, then their cell growth is that daughter cells will grow. Cell growth is taking place, and again the synthesis. That means the cell organelles, DNA replication, everything is happening. Then the preparation. Then that daughter cells is growing. Those cells are growing up. They are getting prepared for the mitotic division, right? So this gap phase, this G2 phase shows the gap phase before one mitotic division, right? This is pre-mitotic gap phase, before mitotic. Just prior to that mitotic division, one gap phase is coming, that is pre-mitotic gap phase or G2 phase. Okay, this is pre-mitotic, which one? G2, and G1 is post-mitotic gap phase. Okay, then here we need to mention about checkpoints. G1, S, G2. This makes the interphase. And here some checkpoints are there. Checkpoints which are considered to check cells, mass and state of DNA replication, DNA damage, all these things. Okay, so during checkpoints, what and all we'll check? These are the points or the checkpoints where we need to consider to check the cell's mass. Then the state of DNA replication. DNA damage. Okay, so all these are getting checked at specific points in a cell cycle. Then the cells are allowed to go past at this such a point only if all the necessary conditions are satisfied. Otherwise, the cell cycle will get stopped. Okay, see here, the cells, these checkpoints means in all these points, cells mass, the state of DNA replication, then DNA damage, anything is there, it is getting checked in few checkpoints. Okay, if everything is satisfactory, then only it will move to the next stage. Otherwise, there the cell cycle, that cell cycle process will get stopped. Okay, it is getting stopped there. Clear? See here we said interphase. Interphase, we know that here there is no division. You can see here there is no division. Division is only in the mitotic phase. So interphase is the non-dividing phase. Interphase is non-dividing phase. So in interphase, we know that there is G1 phase, S phase, and G2 phase. So G1 phase, let's first talk about the G1 phase. In G1 phase, here maximum growth. This is the period of maximum growth. Okay, here we have given cell growth is taking place in G1 phase. That's why I said this diagram you should learn very clearly. Almost all the informations are there in this diagram. Here in G1 phase, maximum growth is taking place and the cell is getting prepared itself for DNA replication. Next stage is what? Next stage is synthesis phase. Synthesis means DNA synthesis. That's about the DNA synthesis. So the cell is getting ready for DNA synthesis. Prepares for DNA synthesis. The cell is getting ready for DNA synthesis and DNA replication. Then here in the G1 phase, RNA and protein synthesis. RNA and protein synthesis is taking place during G1 phase. Okay, so in G1 phase, RNA and protein synthesis is taking place, and the duration of the G1 phase, that is around 11 hours. Okay, here we said checkpoints are there. Here the first checkpoint, that is called as G1 cyclin, and that is between G1 and S. See, after G1, where it moves to the synthetic phase or S phase. So here first checkpoint comes between G1 and S phase, and this is called as G1 cyclin. So that is lying between G1 and S phase, and here the decision about the entry, okay, whether to enter to the next phase or there we should get stopped, there the cell cycle is getting stopped, that decision is made in that checkpoint, whether to go to S phase or that should continue in inert phase. So that decision, it is between G1 and S. Here the decision about the entry in G0, or here you can see G0 or the S stage, that is made during this checkpoint. Okay, and here next, if everything is fine, definitely the cell will move to the S phase, right? See here, each and every point maximum cell growth is taking place in which stage? G1 stage, and that prepares for the DNA synthesis. In G1 phase, what is happening? Preparation for DNA synthesis. And RNA and protein synthesis is taking place during which stage? G1, S or G2. In G1 phase, RNA synthesis and protein synthesis is taking place, and the duration, that is 11 hours, and here checkpoint is there between G1 and S, which is called as G1 cycling. So these many questions, all these are important questions. Okay, previous year questions. Okay, so all these are previous year questions. So you should remember each and every point in this chapter. Clear? So next we are moving to see everything is fine. The cell is moving to next stage. If it is not there, it won't move. It won't cross the checkpoint. Clear? So next we are moving to this S phase. In this S phase, DNA replication and overall amount of DNA doubles per cell in this one. Okay, so here DNA replication is taking place. Which one? In S phase. S phase is a synthetic phase. So here DNA synthesis is taking place. Replication means it's becoming double, right? So it's getting new strands are getting synthesized. That's how replication is happening. So here DNA replication is taking place and new strands of DNA that is getting synthesized. So that is called a synthetic phase. Okay, phase of synthesis. So the amount of DNA will become double, right? Double DNA content. The DNA content will become double. Why it is becoming double? Because the cell is going to divide, then it is becoming two cells. So the DNA content should be the same after division, right? As the parent cell, equal DNA content should be there in daughter cells also, right? Then only the genomic condition will be the same in both the cells, in the parent cell and in the daughter cells. That genetic content should be the same, right? Otherwise, here 13 chromosomes, there it is becoming, or here in the parent cell it is 14 chromosomes, there it is becoming 7, means it will become some other organism. The genetic condition, whole genetic constitution will change, right? So in order to prevent that, here DNA replication is taking place. DNA replication means it's becoming double DNA content is getting synthesized, and these chromosomes, they'll prepare themselves for equitable distribution. Okay, chromosomes are getting ready for equal division. Okay, that should get distributed equally. Okay, and in animal cell, duplication of centriole taking place in this phase. Okay, centriole duplication. Centriole duplication is taking place in this phase in animal cells. While learning about cell organelles, we learned about centriole also, right? So the centriole duplication in animal cell is taking place in this stage, in S stage. Okay, and duration of S phase, that's around 7 hours. Okay, so here the points, here in S phase or synthesis phase, what is happening? DNA replication is taking place, and DNA replication in the sense that DNA is getting double DNA content will be there, and here chromosomes are getting ready for equal division, and centriole duplication is taking place in the case of animal cells. And what is the duration? It takes around 7 hours. Clear? This all these points which I am explaining for each stage, all these are previous questions. Previous year questions. You can see in the question paper, previous year question paper, you can see all these points. Okay. So here S phase over. Next, to which stage it is moving? In this also, as per the demand of the cell division, everything is fine means the cell will move to the next stage. right? So here DNA replication is taking place in the synthesis phase. The DNA synthesis is taking place, then the chromosomes are getting ready for equal division, and it's moving without any issue. Means definitely the cell will go to the next stage, which is the G2 phase. G2 phase. Okay, here G2 phase means synthesis where it will get over. Here DNA synthesis is taking place in which stage? S stage. So that synthesis will get stopped. Synthesis of DNA stops in G2 phase, in gap phase 2. Okay, but still the cell growth continues. Cell growth that continues. Here the protein formation and RNA and protein that continues in which stage? In G2 phase also. And the duration, here the duration 2 to 5 hours it may take. Which one? The duration of this G2 phase. Clear? Here the second checkpoint comes. Second checkpoint, that is mitotic cycling, that lies between G2 and M phase. G2 phase and M phase, the second checkpoint. And here this mitotic cycling that causes the transition from G2 to M phase. Okay, so first checkpoint is here, whether it should move, that is between G1 and S phase. Right. Second checkpoint is G2 and M phase. Here everything is fine, then only it will go to the division stage, G2 phase and M phase. Okay, so the second checkpoint, that is between G2 and M. Okay, and this point, that is mitotic cycling. Mitotic cycling. Okay, the second checkpoint. So here you learned about two checkpoints. That checkpoints that are very important. Both the checkpoints are very important questions. You should know between which this checkpoint lies. Okay, means that G1 to S and G2 to M. The second checkpoint, which is the second checkpoint, which is the first checkpoint, and first checkpoint is G1 cycling, and second one is mitotic cycling. Okay, these are the checkpoints. Another information, that is about the quiescent stage. Quiescent stage. Quiescent stage. Quiescent stage, or we can call as G0 phase. Okay. See, in some cells, like for example, we can say nerve cells and all, nerve cells, then the erythrocyte, they do not divide further during the cell cycle. Okay. Examples, erythrocytes and nerve cells. See, they will not divide further during the cell cycle. So what they will do? They do not divide. Okay. So instead of that, these will exit from the G1 phase to enter into a very inactive stage. exit from G1 phase to which one? To inactive stage. And that inactive stage is called as quiescent stage or G0 phase of the cell cycle. So cells in G0 phase, they'll remain metabolically active, but they will not undergo division or the proliferation that will not take place. They will be metabolically active. This G0 cells which are in G0 stage, that will be metabolically active. But in terms of growth, they do not show any proliferation. Okay, that we need to mention. We have to mention that. So some cells, they are some cells like nerve cells. This is also one previous year question. Which one? Examples of such type of cells which is quitting from the stage of growth. So some cells like nerve cells and erythrocytes, they do not divide, and what they will do instead of that? This is also important question. This is also important question. Okay, and they'll exit from the G1 phase and they will enter directly into an inactive stage or G0 stage. During this stage, this inactive stage is called as quiescent stage, and during this quiescent stage or G0 stage also, the cells will become metabolically active, but they won't show any growth or they won't proliferate. So that is about interphase. Now we have learned about the interphase, right? Next, another phase here, you can see the yellow color which we have marked there. That is the cell division stage. That is the division stage or M phase or the mitotic phase. See, in mitotic phase or M phase, here we have marked this is the mitotic phase, only this small phase. Okay, so here the M phase, this is the phase of actual cell division. Okay, the other phase, the interphase, is a preparatory stage. So this M phase is the actual cell division stage, and that is starting with the division of nucleus. Okay, so how it will start? First, nuclear division. Nuclear division, that is called as karyokinesis. Next comes the separation of, see, the karyokinesis or the nuclear division, and that is followed by the separation of daughter chromosome, and that terminates with the cytoplasmic division. Okay, next is the cytoplasmic division. That means during this, cytoplasm is also getting separated, along with that the genetic material or the chromosome, that is also getting separate. Division of chromosome is also taking place, and that end up with the cytoplasmic division. Okay, completely the cells, the daughter cell formation is happening, or that is ending with the cytoplasmic division. Clear? So this cytoplasmic division, that is called as cytokinesis. Okay, so here karyokinesis and cytokinesis is taking place. Okay, now we need to learn about the karyokinesis. Okay, so here karyokinesis that involves the division of the nucleus. What is karyokinesis? Division of nucleus, right? That is karyokinesis. And in karyokinesis, nucleus can divide, or that can divided either through the mitosis or through meiosis. Okay, so karyokinesis or nuclear division, that is of two types, mitosis and meiosis. Both we need to learn. So one by one we'll learn what is mitosis and what is meiosis. So here in mitosis, we can say mitosis, this is divided into four phases. Which are the four phases? Prophase, metaphase, anaphase, and telophase. So these are the four stages or four phases of mitosis. Prophase, metaphase, anaphase, and telophase. And this prophase, we'll say one by one. What is prophase? Here you can see this is the prophase. Prophase is the longest phase of division. Longest one. Longest phase of this division, that is the prophase. And here you can see the chromosomal material or the chromatin material that is condensing, and condensation of chromosomal material starts from which stage? Prophase. Condensation is taking place, and in animal cells that centriole, in animal cells centriole will be present, so that centrioles will move to opposite poles of cells, move to opposite poles. First in this nucleolus, then the nuclear envelope, everything is getting disappeared. Nuclear envelope, both are getting disappeared in this, or that will go, both nuclear envelope and nucleolus will go off. Then microtubules, we learned about the cell organelles. In that I have said about microtubules and nucleolus and all. So here the microtubules, they are assembled to form mitotic spindle. So spindle formation, in that we said about microtubule, role of microtubule in spindle formation. So here nucleolus disappears, then the nuclear envelope disappears. So here the microtubules, they will assemble and they will form mitotic spindle. So which stage mitotic spindle is forming? During prophase. Okay, so here you can see in prophase, these are the aster, and here the centrioles will be there, and from the centriole the spindle apparatus that is forming. Okay, and here the nuclear envelope will be there, slowly that also will disappear. The nucleolus will be there, then pair of chromatids, you can see the pair of chromatids here. And here you can see the centromere. Okay, centromere in between these two chromosomes. So pair of chromatids you can see, centromere you can see. In which stage? Prophase. Next is metaphase. Here you can see the second stage, that is metaphase, and the arrangement of chromosomes at equator. Okay, so at the equator or at the metaphase plate, with the help of the spindle fibers, or we can say at the equator of that cell, the chromosomes are getting arranged. You can see the chromosomes are getting arranged with the help of the spindle fibers exactly at the equators. Arrangement of chromosome. At equator, that is called as metaphase. Okay, here you can see the spindle fibers, the attachment of spindle fibers to kinetochore of chromosome. So to the kinetochore of chromosome, the spindles are attached. See, in this stage, this is the best stage to observe the shape of that chromosome, size of that chromosome, the number of chromosomes. In this, it will be arranged at the equator in a very clear way. You can see the size, shape, and number of chromosomes. This is the best stage to watch all these things. Okay, that is the metaphase. Next, third comes the anaphase. See, this is the anaphase. Here you can see the third one, that is the anaphase. Here, the centromere, spindles are attached here, you can see. So that centromere is getting splitted. So splitting of centromere. Okay, then after splitting, what will happen? Chromatids, these chromatids will move towards opposite poles. Chromatids to opposite poles. Here you can see the spindles nicely got attached to the centromere. Then those chromatids, this spindles will pull these chromatids towards opposite poles. You can see the attachment of spindles. So the chromatids are moving towards the opposite poles with the help of the spindle fibers. Okay, then this phase, this anaphase, this is the shortest duration phase. Shortest duration. Okay, so that will take only 2 to 3 minutes. 2 to 3 minutes only this phase will be there. Okay, and this is very rapid, very rapid. All these things are taking place. So this is a rapid stage. Which one? Anaphase. And now finally, to which stage it is moving? It is moving to the telophase. Right? Here you can see the telophase, and here the chromosomes. Again, here the condensed one that will become loosened and that will form long fine filaments. Right, and the spindle fibers, here you can see up to the third anaphase, you can see the spindle fibers. Here spindle fibers are disappearing, right? And envelope nuclear envelope that reforms around the chromosomes, it is getting reformed, right? Because it is becoming two separate cells after division, right? Chromosomes reached. So now what should happen? This chromosomes, everything got separated. That duplicated chromosomes got separated. So now that got two equal sets of chromosomes. Now what should happen? That should become two separate cells, right? So what and all should be there to become a separate cell? Nuclear envelope should be there. Here inside that chromosome, one set should be there. Then reappearance of nucleolus, Golgi complex, all these endoplasmic reticulum, everything should get separate, right? Cell organelles also formed, new cell organelles formed. So all those should get separated to make it as a separate daughter cell. So here nucleus, that is all completely, the nucleus is getting divided, all the materials which are present inside the nucleus, everything is getting divided equally, and it is getting equally distributed into two daughter cells. Got it? So here you can see pair of centrioles, nucleolus, chromatin threads. Again this condensed chromosome, it will become thread shaped, it will become loosened and it will become thread shaped. Okay, then here you can see the cytokinesis. Okay, the first step karyokinesis is this one, right? After the nuclear division or the karyokinesis, cytoplasmic division also will take place. And here we need to tell about mitotic poison. Mitotic poison. What is that? Mitotic poison or colchicine. Colchicine or mitotic poison. Actually, this colchicine or mitotic poison, this is an alkaloid. Okay. So all these stages are very important. The things which are happening in each stage, that is very important. See, you can see many questions in the previous year question papers from this portion, cell division, and here the mitotic poison, which is also a very important previous year question. So this colchicine or mitotic poison, that is an alkaloid. Okay, that's one question, alkaloid. Okay, and that is extracted, this colchicine is extracted from colchicum autumnale. Colchicum autumnale, and this will act as what? This will act as a poison for mitosis. Okay, this alkaloid colchicine that will act, which is extracted from colchicum autumnale, from that it is extracted, and this will act as a mitotic poison, that will act as a poison for mitosis. What do you mean by poison for mitosis? That arrests or that stops cell division. This stops cell division. Okay, so cell division, how it is getting stopped? Cell division at metaphase, this stage. Okay, here the cell division is getting stopped. Stop cell division at metaphase. Okay, so what will happen if in metaphase cell division is getting arrested or stopped? What is happening? That causes polyploidy. Okay, in metaphase cell division is getting arrested means what will happen? Polyploidy. Polyploidy will happen. That means multiple sets of chromosomes. Here already an organism will be having single set of chromosomes, right? Here already DNA synthesis is taking place, because that should get distributed for the next newly forming cells. Two sets of equal sets of chromosomes should be ready, right? For that it is making a new set, or it is synthesizing DNA, right? So there the cell division process is getting arrested, or if there it is getting stopped, means in one cell double set of chromosomes will be there, right? So why that double set? Polyploidy, more ploidy will be, or the number of chromosomes, the number of sets of chromosomes will be more, right? So that will change the condition of that cell. Genetic condition of that cell is getting, because it is not going to divide, there it is getting stopped. So that instead of the complete set of chromosome, that cell will be having extra set of chromosomes, right? So that causes polyploidy, and that inhibits this alkaloid colchicine can inhibit the formation of spindle fibers and karyokinases. Here cell division is not taking place. First of all, spindle formation is also not happening there, right? Metaphase here double, it's getting arranged there. DNA synthesis is taking place and it is arranging centrally, but their spindle formation is getting stopped. And after that, spindle formation, it's not pulling towards both the sides. The spindle is not separating the newly synthesized set of chromosomes. That means equal division of chromosomes is not taking place. There no karyokinases, or the cytoplasmic division is not taking place. So what will happen in that stage, in metaphase, the cell is in metaphase, and that will be having more sets of chromosomes. So that causes polyploidy, and karyokinases or cell division is getting stopped at that stage. So that is called as mitotic poison. Poison in the sense it is stopping the cell division. Which one is that? Colchicine, which is an alkaloid which is isolated from colchicum autumnale, that stops cell division in which stage? In the metaphase. Okay, this is important previous year question. This is in metaphase, and that causes that cell division is getting stopped, and that causes the polyploidy. Okay, after this, now after telophase we reached, right? And there is one special or important note which is stopping. Sometimes that may get stopped in some stages. That's what we said. See, after karyokinases, what is that? Next is cytokinases or the cytoplasmic division. Okay, so in this cytoplasmic division, here you can see this occurs in plants and animals in different style. Okay, so in plants, the cytokinases occurs in plants by cell plate formation. So this is a plant cell, you can see. Here, this karyokinases over, nuclear division over. Next, cytoplasm also should get divided. So here what is happening? One cell plate is forming. First, it will start from here. Then it will grow towards both the ends, both the sides of that cell. Okay, so formation of cell plate is taking place in which one? Here you can see cell plate. In which one cell plate formation is taking place? In plant cell. So during that anaphase, in the late stages of anaphase or telophase, we said, which is the telophase, here already separation got over. During the late phases, in the last stages of anaphase and the starting stages of telophase, you can see what? You can see the cell plate formation, and that's how the cells are getting separated. It is making the cell plate formation is getting completed, and then it is becoming two separate daughter cells. Clear? That is in the case of plant cell. But in the case of animal cell, here you can see the process occurs by cell furrows. See, cell plate from the middle, it is starting and it is growing towards both sides. But here in the case of animal cells, how it is forming, how the cytoplasmic division is taking place, animal cells, in that it occurs by the formation of cell furrows. Okay, here you can see the cell furrow or cleavage furrow. From both the sides, the cell is, there is a cleavage or furrow formation for the cell, and slowly it will go, and in the middle portion it will join, and thus both the cells will get separate. Okay, so this process occurs by the cell furrow in the animal cell, and this is taking place at which stage? Metaphase. During metaphase, slowly it will start. Okay, here another thing we need to learn, that is phragmoplast. So what is this? Phragmoplast is the cell plate found by vesicles of endoplasmic reticulum. Cell plate formed by vesicles of endoplasmic reticulum and dictyosomes. Okay, so during which stage? During cytokinesis, cytoplasmic division, this is taking place. The cell plate which is formed by the vesicles of dictyosomes and endoplasmic reticulum during the cytoplasmic division. So these elements will line up at the equator. What is happening or how it is forming? This will line up at the equator during the late anaphase, in the last stages of anaphase, and then it is fusing to form the cell plate. Okay, and after that cell is getting separated and both daughter cells are getting formed. Okay, so this is the karyokinesis and cytokinesis which is taking place in the cell division period. Up to that, this is the division phase. Up to that, the preparation stage which is called as the interphase. Interphase and divisional phase or the mitotic phase both forms a cell cycle. Clear? So these are the processes which is taking place in the cell cycle. In the next class, we'll learn about the cell division, different types of cell division. So now we said the common cell division process. Okay, which and all stages are happening in the preparation stage, what is happening, and what and all preparations a cell is taking place from moving to move from one stage to another stage. That's what we have learned. After preparing everything for a cell division, it is moving to the divisional stage. And during division, how it is taking place, each and every step of division we learned, and where and all the checkpoints are coming, everything we have learned. Right. Next, about cell division, we will learn, which is mitosis and meiosis. That we'll learn the next class. Thank you.